Good evening, ladies and gentlemen, distinguished guests, esteemed members of the academic and scientific community, and my dear students. It's my profound honor and privilege to welcome you all in the fifth lecture of the Atmanirbhar Bharat Nobel Prize Lecture Series 2024. The sub-theme of today's lecture is Nobel Prize in Medicine 2024. The speaker of today's session is Miss Oshin Agarwal ma'am, PGT Biology, International Hindu School. This gathering celebrates not only excellence but also the relentless human pursuit of knowledge, truth, and progress. I feel fortunate to welcome our Honorable President Sir of International Hindu School, Dr. Suman Kumar Mishra, Manager Ma'am, Mrs. Sandhya Mishra Ma'am, Academic Advisor Ma'am, Mrs. Jayanti Samant, Principal Sir, Mr. Santosh Kumar Tiwari, Vice Principal Sir, Mr. Ganesh Shankar Chaturvedi, and Headmistress Ma'am, Mrs. Anupam Srivastav Ma'am. We are privileged to have with us Professor Gyaneshwar Chaube Sir, Department of Zoology, Banaras Hindu University. His work has opened new horizons and has left an enduring impact on both science and society. Now, I would like to invite our respected President Sir to enlighten us with his words.
Thank you very much. I welcome you all. We are going to lay the foundation for many Nobel Prizes today. I call upon the IHS students to forget about becoming a Bayman doctor, Bayman engineer, Bayman collector. If you don't attend the school and just attend coaching, you become Bayman in your foundation. What India needs in Atmanirbhar Bharat is new research, new PhDs. I have done some research. I will make some points. I just want to tell you, when I came to America, before I came, I went to my maternal uncle, and he says, "Why do you want to go to America?" So I told him, because I have done MD medicine from Lucknow, and there is nothing more for me to study here. I want to study perfection. That's why, looking for perfection, I came to America. So we have to do some fundamental changes in India, and I'm going to make a recommendation. I didn't have time to make a slide, but increased funding for research and development. All the companies in India have to, currently we are 0.8% of the GDP. It has to be much more. Israel has 4.5% of the GDP dedicated to research. Then number two is reform in education and talent pipeline. Early talent identification. We have to inspire the young minds. It is the job of the teachers of IHS and other schools to inspire the students to become researchers. STEM literacy, invocation, role models, and mentorship. Number 3 point, retain the talent and reverse the brain drain. I'm sorry to say, but the current political situation is that hardly there'll be any doctors and engineers coming to America. I'm sorry to say that. There was a period when we gone through. So we have to develop our own so that they can get their dreams. I was getting 1,400 rupees a month as an assistant professor at BHU, and I had to move out because many things. So we have to reform the education. And number 3, reverse the brain drain. I remember Mr. Bhatnagar, who was the Vice Chancellor of Banaras Hindu University, he came to my house in Lima, Ohio, looking for Indian talents to tell them to come to BHU. And he says, I'm offering better chances to them than offered here. Then we have to build world class research institutions. The institutional and autonomy is needed. Centers of excellence. Why BHU is not the top? Why Allahabad University is not the top university of India? Why Gorakhpur University is not the top? Why do we have to go through this um, distance learning just to distribute a piece of paper to people? So, number 5, nurture and culture the risk taking and high impact research. Unless you take risk in life, you cannot push ahead. If you just the routine, promote international collaboration. We have to collaborate with other countries more to improve research, enhance scientific communication. We should have World Congress of this topic, that topic in India. Why do we have to go to Europe or to America to attend a world conference? We have to bring them strategic long-term planning. So all of this is very, very fast. I had to give time to uh, Oshin ji to give her talk. I don't want to hog up all the time. But Atmanirbhar Bharat is an economic concept. And it cannot be economically Atmanirbhar unless there is a knowledge growth so that we solve our own problems. We have to, problems are all right there for us to solve. Pollution, it's affecting so much. Delhi is shut down. Population, we have many things. So we have to look at this research. Oshin will talk about how different applications are there. What different applications of microRNA. You guys can take up more research to solve more problems. Lots of problems, lot of diseases. So we have to have a good. I'm very happy that Dr. Chobe is going to join us. And
Sir,
Sir, he has
sir,
joined.
oh, so चौबे जी बहुत बहुत धन्यवाद आपने हम तो ये कहने जा रहे हैं कि आप बीएचयू को नंबर 1 ले आइए वहां के जो पीएचडी निकलते वहां के नहीं सब जगह हिंदुस्तान में यही है कि पीएचडी किसी ने कर लिया तो खाली थीसिस पूरी कर लिया जस्ट कंप्लीट अ थीसिस एक टॉपिक दे दिया और उसमें कभी जाओ स्कूल कभी ना जाओ कोई बात नहीं है अमेरिका में पीएचडी होता है So they put them through intensive training. अगर कोई फिजिक्स में पीएचडी किसी ने किया तो he becomes a master, original thinker of physics. केमिस्ट्री में किसी ने किया तो he becomes an authority in chemistry. ये खाली की पीएचडी का हमारा ये टॉपिक है उसको हमने प्रूव कर लिया this system has to be done with. अब ये हिंदुस्तान को इसकी ज़रूरत नहीं है कि थीसिस लिखो और 1-2 साल में एक पीएचडी के लिए बना दो ये नहीं चाहिए वी डोंट नीड दैट आपको करना है तो फंडामेंटल रिसर्च में लगाइए 4 साल लगाइए 3 साल लगाइए बट प्रोड्यूस समथिंग सो आई डोंट वांट टू take up more time. I like to talk, but thank you very much. And I yield to Oshin Agarwal.
okay, thank you, sir. Just allow me to share my screen. Okay. I hope I am perfectly audible and my screen is visible to everyone.
Yes, ma'am.
Is it so? Okay, thank you
Yes,
so
ma'am.
much. Thank you so much. I'm wishing you a very pleasant evening to everyone. So, welcome to Atmanirbhar Bharat series, an event organized by International Hindu School. So, in the year of 2024, the in the year of 2024, Nobel Prize in Medicine and Physiology was awarded to two scientists, Victor Ambrose and Gary Ruvkun, for their discovery of microRNA and its crucial role in post-transcriptional gene regulation. I know it is a very complex topic, but I will simplify for you all. So moving on to our next topic, this Victor Ambrose and Gary Rufkun, they were actually working on a nematode, a roundworm, which has a transparent body and a very simple organism. They were working on this organism to learn how it regulates its genes. And being transparent in its body, as you can see, the researchers were able to see each and every cell as it develops. So what happened when they were working on Caenorhabditis elegans? They found out two genes, LIN4 and LIN14. LIN4 was a kind of miRNA which they found to be bound to LIN gene's primary transcript, and that's how they found miRNA. They discovered miRNA and also its role in post-transcriptional gene regulation. Imagine this discovery was done in the year of 1992, and they got the Nobel Prize in 2024. And actually, this their groundbreaking discovery has opened new gates to the world of gene regulation. So before taking much time, let's go on. Let us understand first what is microRNA. Micro, as the word is indicating. Micro means tiny. Micro means small. So it is a small class or small type of RNA molecule, and it is a non-coding one. In biology, coding means, in computer coding means, of course, different. And in biology, coding means the one that cannot produce protein. So it is that RNA which cannot produce protein, because the common function of any RNA is actually associated with protein production. mRNA, it translates a protein chain. tRNA, it assembles amino acids. But this microRNA, it does not produce any kind of protein. Second function is it regulates gene expression. Actually, the gist of our PowerPoint presentation lies here. So listen very carefully. Let me first tell you the meaning of word expression, the way we express ourselves through our gestures, through writing, through speaking, through language. In the same way, gene also expresses itself, but here the word expression means synthesis of proteins. Proteins are synthesized by genes in our body. Proteins are responsible for your eye color, for your hair color, for your body weight, even your body complexion. Different traits of your body are governed by the information encoded in this gene. And the process of expressing that information encoded in the genes to make a functional genetic product, which is, of course, protein, is called gene expression. So, in a nutshell, gene expression means formation or synthesis of proteins, which are actually responsible for various traits of our body. मतलब बेसिकली आप ये समझ लीजिए, आपकी आंख का कलर, आपके बालों का कलर, ये सब कुछ प्रोटीन के हाथ में है, in very layman language. And the length of this microRNA is 21 to 25 nucleotides in length, so it is a very short one. And just like I told you, it does not synthesize any protein, but it regulates the synthesis of protein. It itself does not form a protein, but it it acts like a switch, whether it can turn off the gene or slow down the protein production or just turn it off.
It's like a traffic light. It's like a
Yes,
policeman at the
yes.
चौराहा वो बता रहा है इधर से जाओ उधर जाओ.
Yes. So yes, so it tells that gene to just stay shut, and either you slow down or you just don't produce protein at all. Okay, and it regulates by silencing protein synthesis. It regulates genes by silencing protein synthesis. Silencing means shutting down. Shutting down means not making proteins. Okay, so it regulates genes. Regulation means it will switch off the genes. Now, it also forms RNA-induced silencing complex, RISC. Basically, this silencing complex is a complex of proteins like dicer enzymes, argonaute proteins, which actually target the mRNA and degrades it. We will talk about it later, but for now, you just get the idea that it degrades the mRNA so that protein is not formed. Yeah, and of course, it is an epigenetic modulator or it is an epigenetic regulator. Coming on to the next slide. Okay, before I show you working of microRNA, it is very important to understand few basic things about protein production. Firstly, I hope my second slide is visible to everyone. Here I have a picture of a cell, a chromosome, and a DNA. Okay, so look at this picture, everyone. Here is a cell, okay, and inside this cell, there is the presence of a nucleus, and inside this nucleus, there is genetic material, or in very simple words, DNA, right? DNA, chromatin material. Huh. Let us come to this. You can see a rod-like structure, chromosomes. Okay, these chromosomes are basically a very long strand of DNA which is neatly coiled and tightly packed to fit inside a tiny nucleus. Okay, so when DNA is neatly packed and tightly coiled, this long strand of DNA, it turns into a supercoiled structure, thick condensed structure called chromosomes. Chromosomes, you can see it here. Okay, a X-shaped structure on my screen, that is a chromosome. Okay, now come on to the third structure. This is a twisted ladder. ओके एक ट्विस्टेड सीढ़ी जैसा एक लेडर जैसा आपको दिखेगा और आप इमेजिन कीजिए यू हैव टू रोप्स एंड ट्विस्ट देम टुगेदर so this DNA is a twisted ladder like structure, and in this twisted ladder like structure there is the presence of some nitrogenous bases: cytosine, guanine, adenine, and thymine. Okay, very new terms. Let us understand it. आपकी बॉडी की जितनी भी सीक्रेट इंफॉर्मेशन है वो इन नाइट्रोजनस बेसिस में छुपी रहती है. सो दे कैरी और दे एनकोड all the genetic information of your body. Just like I told you, gene synthesizes protein. The information for that particular protein formation is actually decided or is actually hidden or encoded in these nitrogenous bases: cytosine, guanine, adenine, thymine. So all that information for protein synthesis is basically stored in these four letters or four nitrogenous bases. To ease our understanding, these nitrogenous bases are represented as ATGC in DNA and as AUCG in RNA. Basically, this thymine is replaced by uracil in RNA nucleotide. They make up nucleotide. So these are nitrogenous bases which are represented by A, T, G, and C. So the genetic information is stored in the nitrogenous bases of DNA, and then they produce different types of proteins. I hope this much is clear. Moving on to the next slide. Yes, here we have to understand a very basic concept of protein formation. And in biology, this concept is famously referred to as central dogma. DNA replication is also there, but I will just focus on transcription and translation. So these two activities or these two processes are a very important part of central dogma. Okay, so here you can see a cell. Okay, I hope my third slide is visible to everyone. There is this cell, and inside this cell, there is a nucleus. And transcription and translation. First, let me introduce transcription in a very simplified way. To my dear students, I know you all love writing on rough copies in your classroom. You never take your fair copies to classroom. So what you guys do, you jot down in a very hurried writing in your copies, right? You write down everything in your rough copy in your classrooms. And then when you get back home, you rewrite everything perfectly, neatly in your fair copy. तो ट्रांसक्रिप्शन कुछ ऐसा ही है, जैसे आप लोग रफ से फेयर करते हैं, रीराइटिंग करते हैं, ट्रांसक्रिप्शन इज जस्ट लाइक दैट. द आई टोल्ड यू, एटीजीसी में प्रोटीन की बनाने का सीक्रेट है, तो उसी एटीजीसी को दोबारा से लिखते हैं एमआरएनए के स्ट्रैंड पर. ओके, सो बेसिकली transcription is the copying of the nitrogenous bases onto mRNA, or it is just like rewriting of the genetic codes, or it is rewriting of the nitrogenous bases on the mRNA from DNA to mRNA. Okay, so from DNA to mRNA, that is called transcription. So information, the nitrogenous bases which are present on DNA, they will copy the entire thing onto the mRNA, and this mRNA, it will then produce protein. So I hope transcription is clear. It is like rewriting information. It is like rewriting information on mRNA. Now, in case of prokaryotes, there is generally no demarcation into nucleus and cytosol or cytoplasm. So mRNA does not undergo any kind of post-transcriptional modification. But in case of eukaryotes, this mRNA undergoes post-transcriptional modification. mRNA comes out from the nucleus, undergo post-transcriptional modification, and gets ready for the protein synthesis. Okay, so this mRNA, it undergoes translation to for protein. Okay, one more interesting example. When Narendra Modi visits Japan, he does not know Japanese, है ना? And Japanese Prime Minister of Japan, he does not know Hindi. So here a translator works for them, who conveys the language and the messages and makes the communication easy between two PMs. In the same way, the information encoded in the language of ATGC is understood by tRNA, ribosomes, and RNA polymerase, and they make a polypeptide chain. Okay, see this. See this diagram. ATGA, TC, GTA. All these are nitrogenous bases. And now you see how DNA has copied its nitrogenous bases onto the RNA. And now you see how the primary transcript, or this RNA transcript or primary mRNA strand is translated into a polypeptide chain. See these three nitrogenous bases, everyone. A, U, G, all these alphabets. These are not ordinary alphabets, okay? These are special kind of alphabets, or storing information for particular protein in a coded language. Three nitrogenous bases make up a genetic code, and these genetic code are specifically encoding a particular protein. These three letters combine together, and these three letters make up a genetic code that specifies a particular amino acid. And these amino acid, they join one by one, forming a functional polypeptide. And that's how all the proteins of your body are formed. So transcription and translation, these two important activities goes on in every living organism's body for the formation of functional proteins, nitrogenous bases and functional proteins, transcription and translation. I hope it is clear. Coming on to the next slide, working of microRNA. Just like I told you, अब ये माइक्रो आरएनए तो कोई काम करता नहीं है, ये बस स्विच ऑफ करता है जीन का. एंड जस्ट नाउ यू हैव सीन हाउ जीन्स दे सिंथेसाइज और हाउ डीएनए it synthesize a protein. So these are originating from double-stranded RNA precursors, and they convert into mature miRNAs. And I told you, RNA-induced silencing complex, it is a complex of proteins which contain dicer protein, orgonaut protein, and many other proteins. So what happens is miRNA, it it is originated, and miRNA is somehow produced in our body also, and the amount is very specific. If the amount goes increased or decreased, major mishaps can happen in our body too, as they are epigenetic modulators. Now, after transcription has occurred, means after the primary mRNA transcript has been formed, this miRNA just before translation and just after transcription, this miRNA loads itself or attaches itself with the RISC complex, and it then attacks target DNA. Okay. Here I would like to specify one thing. The work of degradation or the work of destroying the primary mRNA transcript before it converts itself into the polypeptide chain, it is done by RISC complex, proteins of RISC complex. Then why? What is the importance of miRNA here? The importance of miRNA is too much. miRNA is that molecule which identifies the target mRNA, which shows the way to RISC, where to go. So it actually identifies the complementary sequence on the target RNA. So degradation work is done by RISC, proteins of RISC, and identification work guiding work is done by miRNA. It actually guides. It acts like a vehicle, and it actually guides this RNA-induced silencing complex to go and degrade the target mRNA. And in this pictorial presentation, you can see again a cell. Inside this, there is the nucleus. There is a presence of mRNA, a primary transcript, which has still not undergone translation. Now, this is the site where miRNA can bound. Now, you can see argonaut proteins, RISC complex. My cursor is moving, please see. This is the RISC complex with miRNA. They would just bind here and then degrade miRNA mRNA transcript, and after degradation, the mRNA transcript is of no use. It will lose its functionality. It will never form proteins. This mechanism is of great use in apoptosis and suppressing tumor genes. And this is called post-transcriptional gene repression. मैंने आपको बताया gene expression means synthesis of protein. Post-transcriptional means after transcription has occurred, means after primary mRNA strand has been formed. After that, the degradation occurs. So it is very much in accordance with our with the title of the discovery, its role in post-transcriptional gene regulation. So this is miRNA and how it degrades the proteins. Next. Okay, epigenetics. It is a very new and an emerging topic, epigenetics. We can change our genes. We can modulate our genes according to the environment we are living in. So it is the study of heritable changes in gene expression without altering the DNA sequence, means epigenetic modification affect cells reading the genes, but it does not alter the sequence of the DNA. The sequence of the DNA will remain the same, but we can actually change the way it reads the genes. And DNA methylation can be done, histone modification. Basically, in this histone modification, the our DNA can either become loose or it can become become very tightly packed by this histone modification. Phosphorylation means addition of protein, and this non-coding RNA regulation is the topic of our presentation only, work of microRNA, and again its role in post-transcriptional gene regulation. This epigenetic genesis means how we can change the coding of the DNA by miRNA. Then we can, the mechanisms by which epigenetics can occur, deactivating the gene, stopping the gene from making a copy, means again no protein synthesis, then stopping the copied gene from producing the protein. And even if the protein is formed, there are chances there are miRNA present which can block the protein function. It either slows down or it just stops the protein function altogether. Then again, breaking up the protein before it can work, means making the protein dysfunctional or denaturation of the protein can occur. So three process: DNA methylation, phosphorylation, histone modification, and again the use of microRNAs. Next, again, environment influences our genetics a lot. The kind of diet we are taking, toxins we are taking, the amount of stress we have, our physical activity, each and everything can influence expression of genes up to certain extent. Let's take the example of two twins, identical twins, monozygotic twins, which are produced after cleavage in the same zygote. These twins, although they share the identical DNA, they have the same DNA. But if you keep both twins in different environments, both twins have different levels of immunity, different levels of intelligence, and difference in the level of social behavior also. So twins which are sharing the same DNA, due to the influence of the environment, can show difference in their traits. So epigenetic modulation can be brought about by diet, psychological state, environment, use of drugs, exposure to chemicals, toxins, and many other things. Again, uh, let us come to the application part of RNA interference or use of microRNA. MicroRNA is again double-stranded RNA, and this diagram it shows how microRNA or this double-stranded RNA or RISC RNA interference has found application in the world of agriculture also. There is a nematode, Melidogyne incognita. It is a roundworm. It it actually infests the roots of tobacco plant, and it reduces the crop yield to some extent. The same concept of miRNA and RISC complex has been exploited here for reducing the infestation of Melidogyne incognita by a technique called RNA interference. Means RNA is interrupting the production of proteins of Melidogyne incognita. So again, the RNA strand is there. The RISC again loads itself along with the miRNA or siRNA. Both are double-stranded proteins. siRNA is same as miRNA. Both belong to the same class of RNAs. And again, they are cleaving this siRNA is cleaving the dsRNA of Malidogyna incognita, and again, it is unable to produce functional proteins. So it has found application in in the world of agriculture. And बायोटेक्नोलॉजी का यहाँ इन्वॉल्वमेंट है क्योंकि we send this we send the genes of siRNA to a vector, Agrobacterium tumefaciens. Again, it has found role in turning off or on, switching on the cells. MicroRNAs involved in epigenetic regulations. They act as tumor suppressor cell. It has it has a wide family of miRNAs. A little bit of amount, a little bit increase in amount of miRNA can be harmful. A little bit decrease in amount can be harmful. It has to be in an optimum amount. So it can switch off the tumor genes or oncogenes, and it can also prevent cancer. Then in the pregnancy, again epigenetics works. The kind of environment a mother is living in affect the behavior of the baby in later life. It affects the development of the baby also. For example, if a mother is taking a diet which is rich in fats, the baby tends to be obese in the later life. So it affects baby's health up to certain extent. So miRNA का भी हाँ बहुत रोल है. miRNA amount actually makes everything. In happy and calm mothers, miRNA is at correct level, but in depressed mother, the levels of miRNA changes, and they also result in anxious and cranky kids. So in a nutshell, microRNAs are like switches which turn off the genes, and they are of great use in research and development. Thank you. That's all from my side, everyone.
Thank you, Oshin ma'am. It was indeed a very insightful presentation. We are privileged to have with us Professor Gyaneshwar Chaube sir from Department of Zoology, Banaras Hindu University. His work has opened new horizons and has left an enduring impact on both science and society. For further insights, I would request Professor Gyaneshwar Chaube sir to share his words of wisdom with us.
Can you hear me?
Yes,
Yes,
sir.
Now
sir.
we can hear it. Now we can hear
Yes.
you.
Oh, great.
Yes, sir.
Mishra sir, namaste. How are you?
Oh, आपके दर्शन का भूखा हूँ.
Oh, very good to see you, sir.
Same here.
Okay, great, Oshina. The presentation was really amazing. I enjoyed each and every bit of your
Thank
slide.
you. Thank you
And
so much, sir.
and the way you presented, that's really fantastic. I mean, a professor or a level of researcher could present this kind of work in a similar way. So congratulations
so
to
much,
you.
sir.
You did a great job.
Thank you
So
so
because
much,
my
sir.
job is to summarize your work, I could add in a nutshell that usually what we see that before the discovery of 2024 when they got the Nobel Prize, everyone was knowing that there is just on and off. So if some gene function, then it is on. If some gene does not work, it is off. But this discovery has brought that genes are not just on or off, but they are fine-tuned by these small, tiny molecular conductors, and they are the microRNA. So how we should correlate with this whole system of life. The thing is, before that, as Sheen has shown that there is just DNA, then DNA to RNA. So the stepwise process is not just only few tiny and few bigger molecules are involved. But as you see that whenever we play some music, we use bass and treble to make the music more useful or more nice, more better hearing by fixing all the control. So as you may also use the various levels of controls. So similarly, the discovery of this microRNA has brought to the global world that, okay, there is not just this is like a black and white process. Okay, there are not just two people, DNA and RNA are involved. There are more, and even so far, 2000 microRNA has been discovered. And with the time, people are, because this has opened a very new avenue. So people are discovering more microRNA. And
Yes, sir.
with this discovery, what they started to think, what they started to learn, that okay, because you see that there is a DNA to RNA, RNA to protein, but why each and every protein makes a different organ, something like then there is a skin, there is a nerve cell, there is nose, there are eyes. Who make give them this coding information that okay, you have to make only eyes, you don't have to make the ears. So these are the molecules, and also by during this discovery, we also learned that because whenever we talk, we say that okay, our 98% of the DNA is junk DNA, only 2% are the functional. But in reality, this 2% is being controlled by the remaining 98%.
Yes.
So things are not so easy, not in black and white. There are many major people in the sense if you say like let's say let's compare that how Banaras is running. So in that sense, not just Banaras mayor is controlling everything. There are several smaller tiny molecules, tiny people who are controlling all the Choraha and everything at the Banaras. So the same way in our body, this microRNA, the discovery of microRNA has brought into our knowledge that it is being tweaked by the as similar thing that uh, we are born, we are being raised by our parents, but when we come to school, we have been shaped by the teacher. So similarly, the teachers are microRNA who are playing a greater role in shaping your future. And usually, you can see that as we also discussed in yesterday talks, that there are just four molecules, A, G, C, and T. And as Oshin said that the thymine changes from uracil and that becomes the RNA. So this is just 22 nucleotide long in AGC
Yes, sir.
and uracil. So such a small molecule and playing such a bigger role in organ formation, in many infection, also in cancer, it has a very greater role. So we see that the things are getting more and more complex as we go deeper, but they are helping us to understand the humanity more and more,
Yes.
to make a better health for everyone, to control the diseases. And junk is not just the junk. I'm not telling about junk food. You have to think about the junk DNA. So junk food is really junk food, as Dr. Saab can tell you more about it. But the junk DNA is not really junk DNA. It controls many part of our activities even. So the as Sheen also said that the mood of an individual changes with the expression of this kind of RNA. So yes, indeed, and see the work it has been published in journal Cell, and it has published in year 1993, but it took a lot of time to consider, to understand the scientific community that this is one of the best work. And so far, we know that 60% of our genes are actually not controlled by just the RNAs. They are controlled by these microRNAs. So far, more than 2000 has been discovered, but slowly more and more is being discovered, very different kind of microRNAs. So whoever, what we feel that even the Indian society that brings the role of each and everyone, as you see that during the marriages, even someone who is making the pots, they are also important because they have to bring the auspicious pot to the home. So similarly, as holistic view, our culture, what we where we are being raised in India. Similarly, the same thing is happening in our body, in our system. So I think this much is enough from my side, and I'm really happy to be here. And I really like the presentation and how Oshin has presented the thing. So thank you very much, everyone. Thank you very much, sir.
Thank you.
Thank you
Thank
so
you,
much,
sir.
sir. Thank you, sir.
Thank you,
It means
sir.
a lot.
Uh,
Yes,
before
ma'am,
you
you go
open,
ahead.
before you open for questions, I want to make some additional comments. I made some, I think, before Chaubeji joined. He missed that part, but it will be in the recording. It's being recorded. I want to say something that don't forget Ashtavakra. I think people who know that story, they know what I'm talking about. Ashtavakra, his father was teaching all the Ved to his mother. And Ashtavakra learnt all that during pregnancy. We have to start building the scientists from pregnancies. I call upon all the mothers to get pregnant and have scientific material fed into them. So we create original scientists. We start learning from inside the womb. Ashtavakra contradicted his father through his mother's mouth. He says, you are not talking, you are talking Dvait, and actually there is Advait. So they cursed him. The father cursed him. He says, oh my God, he's talking already like this. You'll be twisted at 8 places in your body. And later on, it's a very interesting story. Anyway, the second thing I want to tell you, the doors to America have been shut down, practically. So we have to create institutions where we don't have to go to learn. I told this story earlier to the gathering that when somebody asked me, why are you going to America? I said, I want to learn more perfection. So I came to Jocelyn Clinic where only diabetes is taught, nothing else. So I was interested. Anyway, so we have to create institutions of excellence. Number two, I request Chobeji to organize a conference on Mahabharata, and I will give you some funding. I cannot give you the whole, but I'll give you substantial funding to organize this. What can we create new out of Mahabharata? All these weapons they were using, the mantra shakti, we have written it off. Mantra shakti, oh, this is bogus, nobody does it. But mantras had so much. How did they affect? ये research करके नए लोगों को बताना. So I don't want to hog up the time, but last request I have is stop producing these PhDs which you are producing. In America, when a PhD is produced, they become authority on that subject. उसको सारा they taught physics में अगर किसी का हुआ तो उसको सारा 2 साल 3 साल तक दे अटेंड क्लासेस इन फिजिक्स। ये नहीं कि थीसिस लिख दो तुम, पार्ट टाइम कर लो, इंटरनेशनल हिंदू स्कूल में जाके काम कर लो, पार्ट टाइम 4 घंटे के लिए इधर आ जाओ। ये फेक है, दिस इज ऑल बोगस। बंद करिए इसको, इस नाटक को बंद करिए, ये नाटक है। आप लिखिए उसको, जो अथॉरिटी है जो स्कूल को कंट्रोल करती है, जैन साहब से हमारी मीटिंग कराइए, बंद करिए ये सब पीएचडी, और जो एक्चुअल पीएचडी है वो करिए। जो यूनिवर्सिटी ग्रांट्स कमीशन है, आई वाज ए प्रोफेसर देयर एट BHU, आई हैव मेट द ग्रांट्स कमीशन। आप उनको लिखिए कि रियल पीएचडी होना चाहिए, ये फालतू है ये सब। तो आई डोंट वांट टू, I will come and talk to you later when I arrive in Banaras, but you should open it for questions, maybe some children. There are so many opportunities for research, so many problems. You should think no material is waste. Waste तो कुछ होना ही नहीं चाहिए, उसको सबको convert करो. And just remember, kids, knowledge is power. Knowledge is power, not the atom bomb. The knowledge is the power. How come? Israel does some original research and we follow Israel. Look at this. It should be the other way around. We should do original research and everybody should come here to get trained, not to go to America. So I will stop here. I'm full of vim and venom. I'm sorry. Okay, go ahead.
Okay, thank you, sir. So now I would like to request the students to raise some questions. Students, you could give your questions in the chat box because right now there are no questions available.
There are so many applications for disease. This microRNA opens the field. I will tell you, I have in my own practice, I used to have patients who were being treated for cancer and they are developing thyroid disease. So they used to come to me because the RNA was not targeted enough. It was affecting the thyroid also. So there are so many diseases which are affecting. RNA has created a customized prescription. You customize the prescription, just like in Ayurveda. They say you should treat according to the prakriti of a person. If Vata prakriti is more, then increase the pith and the kapha. So customize the medicine. So customize. So many diseases. So there is a lot of opportunity. The kids should get inspired by this lecture.
Okay, I cannot see any questions in the chat box as of now. So I would request the students that you can give the questions to the speakers and we'll be sharing the email address. So you can put up your questions over there. So without further delay, I would like to call our principal sir for the vote of thanks.
Okay. Am I audible to you all?
Yes.
Yes, sir, you are.
Okay. Thank you, Shweta ma'am, for giving me chance to offer my vote of thanks to this entry. Okay. First,
Most
I would like to answer. Yes. First, I would like to extend my thank you to Oshin ma'am. Really, you presented this particular knowledge very rightly, not only rightly, but it was very convincing. A person like me, who is a student of literature, also understood a lot. So definitely, your work was wonderful, and already you have received a lot from the chief guest of the day, Professor Ganeshwar Chaubey, sir. Already I have met him, so definitely we are really very thankful to you, sir, that you again summarized the work of our PGT Oshin Agrawal so nicely, because your name Gyaneshwar, it means you are the master of Gyan, knowledge. So definitely we are very thankful that your this insightful answer explanation, it was really very easy to understand the student like me, who is the student of literature. Third, my thank you goes to our respected president, sir, definitely it is the byproduct of his mind that this type of series, if you are going to telecast Mr. Tiwari in your school, so definitely it would be very beneficial for everyone, for students and those who are going to see this particular video later on. So we are getting recorded this video. So really we are thankful to our Honorable President, sir. Thank you, sir. Next, my thank you goes to Mahasweta ma'am. Really, you moderated this session very nicely, Mahasweta ma'am. So definitely you also deserve our thank you. And
Thank you, sir.
lastly, I would like to thank everyone, those who are now participating and rubbing their shoulders, and you people are really the great treasure of this particular entire session. Without you people, this particular session was not this much meaningful. I offer my thank you to everyone, those who are participating in this program. Thank you, everyone. Jai Hind,
74.
Jai Ram.